Good morning, good afternoon, good evening to our fellow ISCT members listening around the world. I'm thrilled to have another fantastic member spotlight for you this month, hot off the 2025 annual meeting in New Orleans, from which I am still personally reeling. Quick plug that the recordings from that meeting are now posted on the ISCT webpage, and I highly recommend especially the featured presentation from Dr. Applebaum. But today I have the pleasure to be joined by Dr. David Wald. He's a physician scientist at Case Western Reserve University. Dr. Wald is a professor in the Department of Pathology, co-leader of the Immuno-Oncology program at the Case Comprehensive Cancer Center, and associate director of the MSTP program at the School of Medicine. Dr. Wald was also this year's recipient of the Top Abstract Award at the ISCT annual meeting for his abstract entitled Development and Clinical Validation of a Less Than 1 Day CAR T Manufacturing Process. And so this platform represents a potential breakthrough in making CAR T cell therapy faster, safer, and more accessible. So Dr. Wald, congratulations on all this success, and thank you for joining me to talk more about your work.
Thank you. I really appreciate the opportunity to talk to you about our work today. Thank you.
And you continue a long trend it's becoming quite the trend of having first-time meeting attendees win top awards at ISCT. So I understand correctly, this was your first annual meeting ever.
Yeah, that's correct. It was an amazing experience for me. I'm definitely going to come to future meetings after this experience. And I was just really impressed by really the breadth of different areas that were represented from, basic translational research to, clinical manufacturing, clinical trials, regulatory. It really brought together a diverse group of people that you can really learn a lot. And I've benefited quite a bit from my experience.
Oh, we love to hear it. That's great. So we'll look forward to seeing more of your work in the future then. That's awesome. So yeah, let's get into your research. So can you briefly walk us through the motivation behind developing a rapid CAR T manufacturing process and how this relates to broader challenges that we, are facing the CAR T field in terms of accessibility and scalability?
Sure. So this started several years ago, and really our goal was not to make a rapid manufactured CAR T product. It just kind of happened by chance. Initially, several years ago, we were trying to develop strategies to make more efficient, cheaper manufacturing processes in general. And so we kind of developed a very low cost, kind of traditional 10-day process, and then we just kept shortening it, thinking we can save labor costs, save reagent costs by making it shorter, and we just kept shortening and shortening, and all of a sudden it ended up as a 1-day process. So it wasn't really the intention, but it just kind of happened that way during our kind of development to try to make a simpler process.
Nice. Okay. So then can you give us a high level overview of what kind of this product that you made entails, I guess? What are the key scientific or engineering innovations that enabled your team to reduce the time so much?
Sure. I think part of it is not, there's part of it may be the engineering and the specific kind of reagents that we utilize, but I think part of it is just kind of a shift in our kind of understanding of what's required for CAR T therapies. I think initially people may have thought that CAR T therapies were like other drugs where you give more of them and maybe you see a stronger effect in vivo. And now I think more and more groups are realizing that it's maybe more the quality of the cells and not the quantity that may be more important in seeing kind of in vivo effects. And so, our approach is partially we're using a much lower dose. Our current trials utilizing 10 million CAR T cells compared to some products that may use hundreds of millions of CAR T cells, but generating cells that have hopefully better in vivo persistence and durability and hopefully improved side effect profile compared to the more traditional manufactured products.
So, right, actually speaking of that, so your data do suggest that this product has a lower incidence of, or causes a lower incidence of cytokine release syndrome and neurotoxicity. So can you elaborate on how maybe the cell's phenotype or expansion kinetics or something contribute to this improved safety profile?
Sure, and there's still a lot we need to learn to fully understand it. We've treated 15 patients thus far, but at least in those 15 patients, we've seen very little cytokine release syndrome. And in terms of measuring cytokine levels, we see very modest changes in cytokines and also very little neurotoxicity. We think that it's partially due to the fact that we're giving a low dose of CAR T cells. These cells are also immature when they're infused into patients. They're not poised to rapidly kill. They're not differentiated enough that they rapidly kill the tumor cells. And so there is a delayed kinetics of peak expansion of the CAR T cells. It's around 21 days for our median time to peak expansion, which is much slower than with the FDA approved products. And so we think we're seeing a little bit of a slower, gentler killing of the tumor cells, which may be leading to less cytokine production.
It's interesting that some of that, it's almost like you've moved some of that expansion, right, from the in vitro period to the
Right.
or ex vivo period to the in vivo period. That's okay.
Correct. Yeah. Yeah. So really, we're not doing there's no ex vivo expansion. It's just all in vivo expansion.
Gotcha. Okay. Oh, we'll be interested to see if you publish more on the, what phenotype is maybe lending that or what else you learn in the future,
Yeah,
because I think that could help inform a lot of manufacturing across the field, right? Manufacturing processes. That's
definitely.
really cool. So then I also learned that you're transitioning, like in as part of your phase 1B study, you transition from kind of a manual to a semi-automated manufacturing process. And so I wanted to ask, like, what were the main challenges with that, and how did automation impact, these key process parameters that we look at, your consistency, your cost, your scalability?
Sure. So, kind of our initial goal was to develop some kind of robust manufacturing process that could be scalable and used kind of worldwide, even in low middle income countries where cost was a major consideration. Um, and the manual process that we're using, though it was working fine, we didn't view it as very scalable, and we thought that, the consistency of the product may suffer as we try to scale up to reach large numbers of people worldwide. And so we wanted to implement something more automated so it was more controlled, so there's less chances of user error. Um, and so it worked out quite well. I mean, it's semi-automated, so there is a little bit of manual handling, but it's pretty minimized. Now we have a fully closed system. Um, and I think it really helped with the consistency of the product. And so I think it is a lot more consistent. And actually, it requires even less labor, even though it's, a 1 day process. With the current method of production, we have about an hour of user time on 1 day. And then it's a 17 to 20 hour process. So there's an overnight incubation. On the next day, there's about an hour of user time. So one person could conceivably make, up to maybe 5 products in a 24-hour period. With the manual process, it would be kind of one person doing one product. So it's helped with the scalability as well.
Oh yeah, my gears are already turning about, like, workforce staffing and how we could
Yeah.
maximize that process in the lab. That's really cool. Okay. So then, given, that you have this rapid production timeline then with this new process and you have favorable safety and efficacy, how do you envision this technology then changing accessibility to CAR T cell therapies for patients, like you said, in low resource or community hospital settings?
So we've actually recently been contacted by a number of different groups around the world and Latin America, Middle East, India. And so we're working with various partner groups now to try to move this therapy and transfer the technology to other centers and hopefully be able to help the most people in that way. And so we purposely designed it as a pretty simple process so that we could train other centers that may not have huge resources to be able to implement it in their regions and hopefully make low cost CAR T products in that way.
Oh, that would be super cool, especially if you could get a partnership for, supplying all of the necessary, instrumentation and like
Right.
testing infrastructure and all of that.
Yeah. Yeah. So another challenge beyond just the manufacturing, obviously, as you know, is the release testing of the products. And some of these tests are different when you do a rapid manufactured product versus the kind of the traditional manufacturing. And so we've also tried to innovate, the release tests so that we can complement them with the product to make it work efficiently.
We should put in a plug to have someone get you on a roundtable panel next year, or like a concurrent session where we just talk about getting the, regulatory powers that be to approve a testing scheme for rapidly produced products. Because I
Yeah,
think a lot of people are climbing that hill right now, and it would be great to hear.
that would be great. We're happy to participate.
There you go. Heard it here first, guys. I'm going to pitch that to LPC. Okay, so next steps then in your product development. Are you planning to expand to other targets or other disease indications beyond? Currently, you're in relapse refractory and NHL, right? So
Correct.
anything
So,
beyond
so
that?
our kind of lead clinical product is, CD19 CAR based rapid CAR for non-Hodgkin's lymphoma. We also have a myeloma product, BCMA product for myeloma that we just got regulatory approvals. We're anticipating opening the trial probably in about the end of July or early August. So it's coming soon. And then we have a pipeline of solid tumor CAR T products that we're trying to move into the clinic as well. kind of through our group, and then we're also starting partnerships with other groups that have their own, car constructs, and they want to utilize a different manufacturing process. So we're kind of taking both angles.
Oh, that would be great to have other, try out different constructs in this schema, I guess, and see if
Yeah,
it's, broadly translatable, right? Depending on what your vector is.
right.
That's super cool. Okay. Well, we could probably talk for much longer about all of this work. I hope that people will check out your abstract and, any additional publications that you might come out of this. But if people do want to connect with you in the meantime and learn more about what you're doing, how could they do that?
So I guess the best way is to email me at either david.wald@case.edu or david@kurecure.ai. That's the name of our startup company that we're helping to further some of this technology. Those are probably the best ways to reach me.
Awesome. Well, yeah, thank you again for giving me so much time to chat through this, and hopefully we'll speak again soon.
Thank you so much for the opportunity. Take
Take
care.
care. Bye.